Our first major topic today is respiration, and we're going to start with just a review and an introduction. So we're going to go back a little bit, talk about respiration in the broader context, and then introduce the general ideas before we get into some specifics. So the idea with respiration is we need to accomplish gas exchange with the environment. And as you can guess, this is critical for homeostasis. You need to take in things like oxygen and get rid of waste gases like CO2. If you are small and aquatic, you don't need any specialized structure in order to accomplish this. So you look at something like a sponge or a flatworm, members of Platyhelminthes over here. We've talked about this morphology before, how it's flat, how every cell is exposed to either the outside environment or the gastrovascular cavity, so you don't need any specialized structures to circulate materials through that body. It can all just happen by diffusion. But if you are bigger or more complex, then you need a specific respiratory structure to facilitate the gas exchange between the body and the environment. There are a number of different respiratory structures we're going to look at, but they all have similar characteristics. The idea is that they are all adapted for gas exchange, either via the air or within an aqueous or marine environment. So they all share this set of traits. They have thin walls, they have a very large surface area, because if you increase the surface area, you increase the rate of diffusion, therefore exchange. They have to be moist because, again, we're adapted to live in an aqueous environment, so all of our processes have to occur in a moist environment, so the exchange surfaces have to be moist, even if you live on land. And if you have a circulatory system, there are going to be lots of blood vessels associated with those exchange structures. We're going to compare different respiratory structures, and we're going to look at 4 different specific structures. The first is simply the surface of the body. This is the simplest respiratory structure because it's not a specialized structure that's adapted for respiration. It's using a pre-existing structure, the outer covering of the body, and just adding a function to it. So this works for organisms that are very small and have a low metabolic rate. So you're going to be looking at ectotherms, things that are cold-blooded, and things that are not moving all that fast. So earthworms are a really good example of things that exchange across the outer surface of their body. Other examples include amphibians, which are doing it kind of to supplement their lungs, which are not great. One commonality that we see in all of the organisms that use the skin as a respiratory structure is that it has to be moist, even if you live on land, and this creates a challenge because organisms like amphibians are at risk of drying out if they're not at least near a watery environment. This is one reason, in addition to reproduction, why amphibians must live near water for their entire lives, even if they are terrestrial. So using the body surface as an exchange structure is not a great strategy. It needs to be moist all the time, and it doesn't work that great. It's very inefficient because, again, it's not a specialized structure. So what we often see in organisms that exchange directly across the surface is they may have other specialized structures like gills or lungs, as in the case with the amphibians having lungs. The second type of respiratory structure is gills, and we tend to see this in aquatic animals. So here are 3 examples of gill morphologies. The gills themselves are shown in pink. So we have a type of marine segmented worm. We have some crustaceans here. They have kind of internal gills that are inside the exoskeleton. And then we have the sea stars, the echinoderms, that have gills kind of around the circumference of the arms. One thing that we see in common with all of these gills is that they are folded or extended surfaces that increase the surface area for gas exchange. So in the case of this structure, you don't need to know this word, but the structure is parapodium. In the case of the parapodium, they extend away from the body. In the case of the gills, in the crayfish and the sea star, they zigzag back and forth. They kind of extend and wiggle back and forth, and you can see that here, these folded layers. Those folds increase the surface area, which increase the efficiency of exchange, and all of this is happening in a watery environment, either aqueous or marine. Some gills are actually the most efficient exchange structures in terms of respiration because they use a mechanism called countercurrent exchange. This is where the blood and the water flow in opposite directions, which means that the blood is always exposed to water with a higher O2 content than the blood. So for the entire duration of the exchange, oxygen is flowing from the water into the blood. It maximizes the diffusion of oxygen. Here's a representation of how countercurrent exchange works. We're going to look at the side of this exchange structure where the water enters. So on the left here, we have water entering the structure. The water, as it comes in, is highly oxygenated. Now, it's meeting blood that is also highly oxygenated, but the blood is never going to be as oxygenated as this water coming in. So oxygen will immediately diffuse into that already highly oxygenated blood. As the oxygen moves out of the water becomes oxygen depleted. But even all the way at the far end of that exchange structure, where the blood is entering, the blood is completely oxygen depleted at this stage, or deoxygenated. So the water over here still has enough oxygen that it's going to diffuse into that deoxygenated blood. So you have oxygen flowing from the water into the bloodstream across the entire range of this exchange. In contrast, in things like the human lungs, where the blood surrounds the air, they reach an equilibrium and you actually don't get as much oxygen out of the environment as you do with countercurrent exchange in gills. So countercurrent exchange, highly efficient. We see this in some types of gills, like fish, for example, use this type of exchange in their gills. Our third type of respiratory system is the tracheal systems we see in insects. So this is a network of ducts that branch through the body and actually have openings to the outside. So we have tubes called trachea that then branch into narrower tubes called tracheoles, and eventually those are going to branch and branch and branch and get smaller and smaller until every cell is very close to a tracheole and can accomplish gas exchange essentially directly with the atmosphere via these tubes. One benefit of the tracheal system is that you don't need an additional circulatory system for gas exchange. We'll see in humans that you need a circulatory system because the lungs are not connected to everything else. But for insects, the trachea are actually connected to everything else, so you don't need an additional system to facilitate gas exchange. But this comes with a cost. One is that you have these openings to the outside environment, which are particular avenues into the body for diseases. The second problem is that you need to get a high enough volume of air into all these tubes to feed all of the cells. And once you start to have so many cells, the tubes need to be so wide it becomes a problem for the rest of the body. Basically, they start to displace other things in the body. And this is one of the factors that limits the size of insects and other arthropods. And going back to something I mentioned earlier in the semester about how some arthropods were larger like millions of years ago when the oxygen content of the atmosphere was higher, this is one of the reasons those larger body sizes was possible. You got more oxygen into the body using tubes of the same diameter, so they didn't take up too much space in the body. But with less oxygen in the atmosphere, you get less bang for your buck in terms of the volume of these tubes, so you're limited to a smaller body. So along with the weight of the exoskeleton, the tracheal systems are the limits on the size of things like insects and arachnids, which is good. The final respiratory structure that we're going to look at are lungs. These are specialized for respiration above ground, so we primarily find lungs in terrestrial vertebrates. So these are the organisms that are living on land and conducting gas exchange with the atmosphere, and the lungs are the structure that is bringing the air into the body and removing it from the body. Rather than focus on lungs right now, we're just going to have our own section on how mammals do respiration, which is going to be called mammalian respiration. In this section, we're going to look at the structure of the respiratory system, and we'll also look at the mechanism of respiration in mammals, focusing on humans, of course. And one thing to keep in mind as we talk about this system is that the lungs are not in direct contact with the rest of the body. So this is different from the tracheal systems we just saw, like in insects. In mammals, the lungs are in a specific location and you only conduct gas exchange in that specific location, and then you need a circulatory system to move those gases through the body. So next time we're going to talk about circulation, and we'll talk about that other step in the process. But just keep in mind that this is not the complete system as far as gas exchange goes. You also need circulation in order to complete the process of gas exchange in mammals. Here is an overview of the structures involved in mammalian respiration. We're going to start at the front end, the things that interact with the atmosphere, and then we'll go down and down. So first we have the nasal cavity, because your nose is actually the respiratory structure, whereas your mouth, that we've already talked about, is the feeding structure, even though they kind of connect in the pharynx. So we've already talked about the pharynx before. This is that crossroads between feeding and respiration. Below the pharynx, we have the larynx right here. This is also called the voice box because this is where your vocal cords are located. And the larynx leads to the trachea, which is the large tube that connects the upper part of the respiratory system to the lungs. The trachea has cartilaginous rings, which you can actually feel. So you feel on the front of your neck, you can feel those rigid structures. Those are actually the cartilaginous rings of the trachea. The function there is to keep the trachea open all the time. Now, the esophagus is mostly going to be smushed flat as long as there's not food moving through it. When you swallow, that bolus moves through it and it opens up kind of in a wave-like pattern. But the trachea, we want to be able to breathe all the time. So the trachea is always open. It's the cartilaginous rings that accomplish that function. Below the trachea, it splits into large tubes called bronchi. The singular is bronchus. So we have bronchi in either lung, and then those further split into smaller tubes called bronchioles. But I'm not going to highlight the word bronchiole. An important thing to realize about all of these structures is they are not accomplishing gas exchange. These are just the tubes that the air is going to flow through in order to get to the sites of gas exchange. So now we're all the way down in the lungs. The lungs themselves are a pair of organs. They're spongy and elastic, and they're found within the thoracic cavity, which is a fancy way of saying the chest cavity. Basically, you have your diaphragm right here. Everything above that is the thoracic cavity. I don't know if that's the technical definition, but that's how I think about it. So we have our lungs, and within the lungs, the structures that are actually involved in gas exchange are alveoli. And these are, we've heard this word before, and it means the same thing. These are just tiny air sacs. So it's similar to those little vesicles that we talked about in terms of their morphology. It's just their little sacs of tissue in your lungs. They are surrounded by a single layer of cells, right? So they look like the alveoli we've talked about before, but these are cellular rather than membrane. And that single layer of cells is important because this is where exchange occurs. So you need to have very thin surfaces there. This single layer of cells is surrounded by capillaries, which is what we see in purple right here. So the air is going to flow in, it's going to inflate the alveoli, and gas exchange will occur between the alveoli and the capillaries that surround them. An important thing to keep in mind here is the enormous surface area. The surface area of the alveoli in your lungs is more than 100 square meters for the average person, more than 100 square meters. This is approximately 50 times the surface area of human skin. So this is an enormous surface area over which gas exchange is occurring. Now, as I mentioned before when we talked about the countercurrent exchange, alveoli are not the most efficient way to accomplish this because the air goes in and it's surrounded by the blood in those capillaries. So eventually you reach an equilibrium point where there is approximately an equal concentration of oxygen in both locations. This is going to limit the degree to which the oxygen is going to move into those capillaries. So while this is not as efficient as countercurrent exchange in gills, we compensate for that by having an enormous surface area over which this is occurring to get enough oxygen for all the stuff we need. The mechanism for breathing is really interesting. The word breathing just refers to the mechanical process of moving air in and out of the lungs. And there are two components to breathing. There's inhalation, which is moving the air inwards, and exhalation, which is moving the air out. And the interesting thing about how mammalian lungs work is that this process is achieved through negative pressure. So we are drawing air in rather than forcing it in, is kind of the way I think about it. We use muscles to accomplish this, the main muscle being the diaphragm, the large muscle that sits just below the lungs. And I have to issue a correction here. The diaphragm is a skeletal muscle, even though it's mostly under autonomic control. At some point, I said that the diaphragm was an example of a smooth muscle. That was incorrect. So I apologize. The diaphragm is a skeletal muscle, but it's unique among skeletal muscles in that it's actually under autonomic control most of the time. Although, as you know, you can manually kind of take over control of your breathing. So now let's look at the processes of inhalation and exhalation. So what's going on here with inhalation? We are going to contract muscles during this step, and I want to make that very clear. Inhalation involves muscle contraction. Exhalation is going to involve muscle relaxation, so just be aware of that. So when we inhale, the diaphragm is going to contract. Now the diaphragm is like dome-shaped or arch-shaped. So when it's relaxed, it's longer, right? Muscles that are relaxed are longer. So you have this dome-shaped muscle. When it's relaxed, it's longer. When it contracts, it becomes shorter. Now the two ends of the muscle, the bottoms here and here, those are anchored in place. So when you contract the diaphragm, the distance between them doesn't shrink, but the length of the muscle itself gets shorter, so it moves downwards, right? So it starts off like this big, deep arch, and it becomes shorter. So it moves downwards. At the same time, the muscles in between your ribs are also contracting, and that's pulling your rib cage upwards and outwards. As you contract those muscles, it moves up and out. So we have the upper surface moving up and out, the lower surface moving downwards, and that increases the volume of your lungs. Now think back to chemistry class, you probably talked about this, the relationship between pressure and volume. If you increase the volume, what happens to the pressure in your lungs? Yeah, exactly. You decrease the pressure, right? You increase the volume, decrease the pressure. So now you've decreased the pressure in your lungs. In particular, you've decreased it relative to the atmosphere. So now that's going to cause the air from the atmosphere to move from the high pressure atmosphere into the relatively low pressure lungs. So this is how we inhale. We contract our muscles, that increases the volume of the lungs, causing air to rush in. Exhalation works the opposite way. And the thing about exhalation is that this is the passive part of the process. This involves muscle relaxation. So we relax those muscles associated with the rib cage. We bring that surface down. We relax the diaphragm so it can expand, meaning it moves upwards. So those two things combined, the ribs and the diaphragm, they shrink the volume of the lungs. That increases the pressure, and in particular, increases it relative to the atmosphere. So now air moves from the high pressure lungs outwards into the low pressure atmosphere. We consider this part of the process passive because it does not involve the expenditure of ATP. You're not contracting muscles in order to exhale, you are relaxing them. To get a nice view of how this works, we can go back and forth between these two figures and see how the different structures change during inhalation and exhalation. So here, during inhalation, you can see the idea is contract muscles, increase volume, and during exhalation, it's relax those same muscles and decrease the volume. This is most apparent here looking at the diaphragm. So we have in, out, in, out. The idea is with inhalation, increase the volume by muscle contraction. Exhalation, decrease the volume by muscle relaxation. When you increase the volume, you decrease the pressure, air rushes in. When you decrease the volume, you increase the pressure, air rushes out. That is the process of mammalian respiration. The last thing we'll talk about in this section is how we regulate breathing, and this is super cool. The rate of breathing is actually based on CO2 levels. So this is something that our brain controls on its own most of the time. We don't have to consciously be thinking about breathing all the time, which is really good because one, if we're out and about doing stuff, we have other things to think about. We don't want to have to worry about breathing. But two, if we had to think about breathing all of the time, we had to do it consciously, how would we sleep? Yeah, that would be really weird. We wouldn't be able to sleep, would we? So it's really important that breathing is under autonomic control most of the time. The thing that our body uses to regulate this rate of breathing is the pH of the blood and the cerebrospinal fluid. This is important because that pH indicates the CO2 concentration in our body. You do not have to know this chemical reaction right here, but the general idea is that we take CO2, when our cells generate CO2 as a byproduct of respiration, and it goes into the aqueous fluids in our body, right? Blood, cerebrospinal fluid. That generates carbonic acid, which is acidic. So it dissociates and it releases protons into solution. So that decreases the pH of various liquids in our body. When we are experiencing a higher metabolic rate, we generate more CO2, and that's going to lower the pH of our blood and our cerebrospinal fluid. This is detected by the medulla oblongata. Remember, that's that very kind of ancestral lizard brain part of our brain in the brainstem. So the medulla is going to detect this decrease in pH and say, aha, we have too much CO2. We need to increase the rate and depth of breathing in order to get rid of that excess CO2. That will bring the pH back into the normal range. So the really cool thing about this is the question, why do we breathe? You think, well, why do we breathe? Well, we breathe because we need oxygen. We breathe in order to get enough oxygen. But physiologically, the reason why we breathe is actually to get rid of CO2. That is the reason we are breathing. In terms of like how our body thinks about this, we're not detecting the oxygen. It's all about getting rid of the waste products, the CO2, from cellular respiration. So if anybody asks you, why do we breathe? The answer I want you to give is to get rid of CO2. Isn't that cool? I think that's really cool. Here is an overview of this mechanism. So this looks a lot like those negative feedback diagrams we've seen before, because this is negative feedback regulation. We have our normal blood pH, and if we have an increase in CO2, like when exercising, that's going to cause that pH to fall. So the stimulus is a lower pH. The response is to breathe faster and breathe more deeply, and that's going to get rid of the excess CO2, bringing the pH back into the normal range. So the response brings up the pH. So we have a stimulus going in one direction and a response going the other direction. That is negative feedback regulation to maintain homeostasis, just like we've seen before with lots of other systems. So let's summarize respiration. We looked at a number of different respiratory structures. We looked at the skin, gills, tracheal tubes, and lungs. We looked at the mechanism of how mammalian lungs work. And we looked at how breathing is regulated via negative feedback related to pH in your fluids due to CO2 from cellular respiration. Now that we've covered respiration, we can change gears a little bit. We're going to spend the rest of today talking about excretion.